Phlebotomy National Exam Practice. Let's get started. Which test requires chilling the specimen immediately after collection? Is it A, CBC? Or B, ammonia? Could it be C, lipid panel? Or lastly D, hemoglobin A1C? The correct answer is B. Here is why. Ammonia is highly unstable at room temperature due to ongoing metabolism by blood cells. Placing the sample in an ice water slurry immediately after collection slows this degradation. A. CBC samples are stable at room temperature. C. Lipid panels are typically processed at room temperature or refrigerated after centrifugation. D. Hemoglobin A1C is very stable at room temperature for days. Next question. What is the term for a small, pinpoint red spot caused by a tourniquet that is too tight? Is it A, ecchymosis? Or B, petechiae? Could it be C, hematoma? Or lastly D, cyanosis? The correct answer is B. Here is why. Petechiae are tiny, non-blanching hemorrhages caused by the rupture of capillaries, often due to increased venous pressure from an overly tight or prolonged tourniquet application. A. Ecchymosis is a larger, bruise-like area of bleeding. C. Hematoma is a raised collection of blood. D. Cyanosis is a bluish discoloration from lack of oxygen. Next question. For a newborn screening test, like for PKU, blood is collected onto. Is it A, a lavender top microtainer? Or B, a special filter paper card? Could it be C, a red top serum tube? Or lastly D, a gray top tube? The correct answer is B. Here is why. Newborn screening tests for metabolic disorders require capillary blood to be spotted onto specific circles on a filter paper card, which is then dried and sent to a specialized lab. A, C, D. These tube types are not used for standard newborn screening card tests. Next question. The chain of custody is a procedure used for. Is it A, blood cultures? Or B, forensic or legal drug/alcohol testing? Could it be C, critical care testing? Or lastly D, organ transplant compatibility testing? The correct answer is B. Here is why. Chain of custody is a legal protocol that documents the handling, transfer, and storage of a specimen from collection to reporting to ensure its integrity and admissibility in court. A. Blood cultures require a septic technique but not legal documentation. C. Critical care tests are stat but do not require chain of custody. D. Cross-matching for transplants is critical but follows different protocols. Next question. Which is a CLSI recommended step for site cleaning? Is it A, scrub back and forth vigorously for 5 seconds? Or B, clean in a circular motion from the center outward and allow to air dry? Could it be C, blow on the site to dry the alcohol faster? Or lastly D, wipe away the alcohol with a gauze pad before it dries? The correct answer is B. Here is why. Cleaning from the center outward with friction, using a circular motion, and allowing the antiseptic to air dry maximizes microbial kill and prevents recontamination. A. The motion should be concentric circles, not back and forth, and the time is usually 30 seconds for alcohol. C. Blowing introduces airborne contaminants. D. Wiping negates the contact time needed for the antiseptic to work. Next question. Which additive can cause falsely decreased calcium levels? Is it A, heparin? Or B, EDTA? Could it be C, sodium citrate? Or lastly D, sodium fluoride? The correct answer is C. Here is why. Sodium citrate binds calcium ions. If a citrate tube is used erroneously for a calcium test, or if there is carryover into a serum tube, it will chelate calcium and produce a falsely low result. A. Heparin does not typically interfere with calcium measurement. B. EDTA also binds calcium, but samples in EDTA tubes are not typically used for calcium testing. D. Sodium fluoride does not significantly affect calcium. That's it for today. How many did you score? Let me know in the comments. Please like and subscribe, and tell me what topic I should cover next. Bye.